So today we are going to learn the structure of HIV virus and its life cycle as well, right? That how it causes the infection. Now, the structure of HIV virus you guys have already learned when you guys were in.
Sir, your mic muted.
Okay, what is the structure of this HIV virus you guys have already learned when you guys were in O levels, right? Grade 10. Now, this HIV virus, this is an enveloped virus. This is an enveloped virus. Now, the question is, what is the meaning of enveloped virus? A virus that has the cell membrane of its host cell that is called as an enveloped virus, right? Now, second thing, that this HIV virus, this has two strands of RNAs. So it means that it is an RNA virus. Another thing, that this virus contains 3 enzymes already present in it. Now, here, this is what an HIV virus appears like. Now let's label this. First of all, this over here, this is the lipid bilayer. We cannot call it as a cell membrane because viruses are not cells, right? Now this lipid bilayer is actually the cell membrane of the host cell, which it took.
Checking with the paper?
No,
Result?
not now.
Not?
Right now, class.
Ready.
Right, guys? So lipid bilayer, we call it as lipid bilayer. Next, this blue line which I drew, this blue line is made up of matrix proteins, right? And these spikes which I made, these spikes are actually glycoproteins. And most specifically, this glycoprotein which is outside, this is called as GP120. And its inner part over here, this is called as GP41. Now question is, why is it called as GP120? Why it is called as GP41? Can you think of it? Any reason? Sorry? No. Molecular mass. Right? The mass of this glycoprotein, which is on the outer surface, this is 120, and the mass KDA, right? Kilodaltons. And the mass of the protein, glycoprotein, which is on the inner side, that is 41. 41 KDA, right? Then you guys have learned that all the viruses, they have a protein coat. What is the protein coat of a virus called? Capsid. So this over here, this is the capsid of this virus. Understand? Then this, I told you guys before as well, this is an RNA virus and it has 2 strands of RNA. This is an RNA and this over here is also RNA. And there are 3 enzymes in it. Now, one of the enzymes, this one, let's suppose this is called as reverse transcriptase. A very interesting enzyme. You have learned about transcriptase enzyme, right? Which causes transcription. This is reverse transcriptase. We'll talk about it, right? Then, let's suppose that there is this enzyme, and this enzyme is integrase enzyme. We are going to talk about the function of all these enzymes, and this enzyme over here, this is simply a protease enzyme, right? So this is the structure of an HIV virus. Now let me tell you one thing. HIV causes AIDS, right? Who will tell me the full form of AIDS? Does anyone remember?
Acquired immunodeficiency syndrome.
Very good. Acquired immunodeficiency syndrome. Now here the emphasis is on the word syndrome. AIDS itself is not a disease. It's a syndrome. Why we are calling it as a syndrome? Because of this virus, you do not become sick. Because of this virus, you do not become sick. Only what happens that this HIV virus, human immunodeficiency virus, HIV's full form is human immunodeficiency virus, it hijacks your lymphocytes. And most specifically T lymphocytes. And which T lymphocytes? T4 lymphocytes. Right? Now when it will hijack the machinery of T4 lymphocytes, they will not be able to function properly. So your immune system becomes inactive. When the immune system becomes inactive, what will happen? Any other disease can happen. Like, let's suppose that there is a person A, he got infected by HIV virus, right? Now his immune system is hijacked. If you see Vibrio cholerae enters in his body, he will get cholera. No immunity against it. If tuberculosis, you see this Mycobacterium tuberculosis, that bacterium enters in his body, he will get TB, right? There is a pneumonia bacteria enters in his body, he will get pneumonia. So he can get any disease. Now, it means that because of hijacking of the immune system, this did not cause any disease, but he is getting other diseases. Now, what is happening now actually? This is very interesting fact. Now, all the other microorganisms, they are getting opportunity to grow inside the body of that person, right? They are getting opportunity to infect that person. So the diseases that person get, those diseases are called opportunistic diseases. Cholera he got, opportunistic. TB he got, opportunistic. Pneumonia he got, opportunistic. Sore throat he got, opportunistic, right? So those pathogens got the opportunity to grow, right? That's why they are called opportunistic diseases. Now, so this virus, it attacks the T4 lymphocytes. The question is why these particular type of lymphocytes, they are called as T4 lymphocytes. Because you see, why this virus can only attack on the T4 lymphocytes. Why not on the other cells? Because these glycoproteins, this GP120, they only attach to specific receptors present on the surface of T4 lymphocytes. And those receptors are called as CD4 receptors. Now, boys, let's suppose this is what? This is a T4 lymphocyte. We are going to learn whole cycle in this diagram, right? So let's suppose that this is what? This is a T4 lymphocyte. And on the T4 lymphocytes, let's suppose that these are special receptors which are called as CD4 receptors. What are they called? CD4 receptor. Now what will happen that this HIV, this virus will come and it will attach here on the CD4 receptor. Right? Now, we we can draw rest of the structure very quickly. Right? So it has bind to the receptor, which is the CD4 receptor on T4 lymphocyte. Now what will happen? Endocytosis. What will happen? Endocytosis. Now you guys remember, when endocytosis happens, the vesicles membrane become part of the cell. Understand? And when exocytosis happens, at that time the vesicles, they are released, the membrane is reduced, right? So here this will enter by endocytosis. Now I can show on a side in a small diagram how the endocytosis will take place. That when it will bind this, so these are what? Glycoproteins, right? And what has happened, that this capsid and the internal part has entered there into the cytoplasm, right? So when endocytosis will take place, this part, the capsid will enter over here. Got it? Now, before we move further, let me draw the nucleus of the T4 lymphocytes. Now you guys know that the lymphocytes, they have single large nucleus. Do you remember this or not? We have learned that, right? So this is what? This is the nucleus of the T4 lymphocyte, and let's suppose this is the DNA present inside. I am just drawing one molecule to make you understand, right? Remember that there is protease enzyme, right? And capsid is always made up of what? Proteins. So that protease enzyme will digest the capsid and release the contents of the virus. And what are those contents? So what will happen? The capsid is digested and RNA and contents are released in cytoplasm. Now let's suppose that this over here is what? RNA of virus. Now this virus is already carrying reverse transcriptase in it, right? Next step, what will happen? That the reverse transcriptase will bind to this RNA. So this is RNA, and let's suppose that this over here, this is what? The reverse transcriptase. So what do we write over here? Reverse transcriptase binds to RNA. Now if you guys remember, we have learned in detail from DNA to RNA formation, this is called what?
Transcription.
Transcription. Right? So what will be reverse transcription? RNA to DNA. RNA to DNA. So when there will be formation from RNA back to DNA, that will be called as reverse transcription. So the reverse transcriptase enzyme, what does it do? It causes reverse transcription. And what does it do? It makes DNA. Understand? So what we can mention over here, that DNA is made by process of reverse transcription. Now, next step, what happens? That this viral DNA, now we can call it as viral DNA, right? It enters through the nuclear pore into the nucleus, right? Now, this viral DNA, it has entered into the nucleus. Now, what happens? With the help of integrase enzyme, it is integrating, right? You know the meaning of word integrating, adding something into which is already existing. With the help of integrase enzyme, what will happen? That this viral DNA will be added into the host DNA. Into the host DNA. So I can draw the diagram like this. And I can show right? So you guys can see the difference in color over here, right? So I can mention over here that integrase enzyme adds viral DNA into host DNA. Got it, guys? Now, it has become part of the host DNA. Now, next step, what will happen? Next step, transcription will start. Now, the transcription of this DNA will start and many RNA molecules will be made. Many messenger RNA molecules will be made. So, I can show over here. So, what is happening over here? That now transcription will take place and many molecules of messenger RNA of virus are made. We can mention simple RNA as well instead of writing messenger RNA, right? Now what will happen? That these RNA molecules, they will come out, right? And they will come into the cytoplasm. Now what will happen? Translation will start, right? Next step, what will happen? That with these RNA molecules, the ribosomes, they will attach. And translation will start. Translation of RNAs start. Now when the translation will start, what will happen? The parts of the viruses will be start making. Now, what will happen? Let me show, we use the blue color over here. What will happen? That, let's suppose this is what integrase enzyme is being made, right? Let's suppose this is what reverse transcriptase is being made. Not one, many will be made, right? Let's suppose this is protease enzyme is being made, right? Let's suppose that capsids are being made. Understand? So all these things, the virus parts, they will be made by this process, right? Now you guys know that capsid is protein. Enzymes are also proteins. They will all be made. Next step will be what? Assembly of these parts. These parts they will assemble, right? So next what will happen? Assembly of virus parts. Now whenever you buy an iPhone, on the back of the iPhone it is written designed in California, assembled in China, right? So parts are made somewhere else, but they are just joined in China. Same, the parts are made over here and then they are assembled, right? So what will happen? Eventually it will change into this. And remember, these are RNAs are also coming due to transcription, right? So these RNAs, they will also be added, and these enzymes will also be added inside. Now next step, what will happen? Next step, that this will come out by exocytosis. Now we have learned in detail the process of exocytosis, right? So whenever exocytosis happens, some part of the membrane is covering the particle which is coming out. So when this will come out, that will be like this. Right, guys? So what will happen? That this virus will go and attach to more T4 lymphocytes. Understand? Now, just imagine what is happening. That this cell is being hijacked now. It's not performing its own duty of making chemicals to destroy the pathogens, right? Rather, it is making the parts of the virus. Second, what is happening? When the viruses are going out by exocytosis, so part of the cell membrane is going away, right? So what will happen? The cell is becoming smaller and smaller and smaller. Eventually the cell will finish, right? But by that time, many of the viruses, they will be made, right? And they have gone and infected more T4 lymphocytes. Understood, everyone? Now, here, one thing very, very important. Whenever they want to test for the presence of HIV virus in a person, they test that how much amount of, this virus is there. Now, a person can be HIV positive, but he cannot have AIDS. These are two different things. A person can be HIV positive but don't have AIDS. Means that virus is present in the body, right? But it has not caused the disease. Now remember one thing, whenever an HIV virus enters in the body, it causes the flu-like symptoms. The person will have some trouble in the stomach, right? Nausea or some, vomiting, diarrhea-like things, right? So this will happen and it will last for a few days only. After that, incubation period starts. That depends on person to person, and it can last from 2-3 years up till 10 years or 15 years. That the person, whenever tested, will be tested positive, will not show the symptoms of AIDS. But it can turn into the AIDS at any time. We don't know what will trigger this, but the person can change into, AIDS condition. Now, again I told you in the beginning that the person who will be AIDS, who will have AIDS, HIV positive and AIDS, will have opportunistic diseases, right? Now, as there will be opportunistic diseases attacking that person, so the person needs to be treated, right? Now, you guys have learned that we cannot kill viruses. We learn about that as well at the end of this chapter as well. Why we cannot kill viruses? Can anyone tell me?
So they are living cells.
They are not living organisms. First thing. Second, the antibiotics we will talk about, they work by either stopping the production of cell wall or by destroying the cell wall. Right? So they don't have any cell wall. Rather, they don't have any cell membrane. Right? Even you see this one, we are not calling it cell membrane, but what are we calling it as? Lipid bilayer. Right? They don't have a cellular structure. So as they don't have a cellular structure, so we cannot destroy them. So how to treat this? Now, we will talk about that in the next chapter as well, that you may have heard about interferons. Have you heard about these things? No. Always, you see, we can only use the antibodies against these viruses, right? So interferons, they are a type of artificial antibodies. They are expensive drugs which are used as an antiviral therapy, right? So those are being used, the interferons, they are being used. And during the treatment of HIV, one drug or two drugs, they will not work. They call it as cocktail of drugs. What is a cocktail? You see, when you go and buy juice, when it's mixture of 5 or 7 juices, they say that cocktail, right? Or you see, whatever the leftover juices they are left in there, you see fridge, they mix them and they sell them as cocktail, right? So, cocktail of drugs, because you see, it's very, very, you see, difficult to treat this HIV infection. Understand? So this one thing. Now question is, how HIV spreads? Now HIV spreads usually by body fluid mixing together. Now it is thought that, you see, it was spread due to homosexuality in people in Europe or in other countries. And what happens actually, you see, when there is normal intercourse, at that time, you guys have learned in O levels that vagina produces a natural lubricant, right? But when, you see, this homosexuality happens, they, you see, having intercourse through the wrong pathway, there is no natural lubrication. So what happens that the anal, you see, muscles and the rectal muscles, they tear and blood comes out. And from the semen of the infected person, the virus can easily enter into the blood. And it does not mean that, you see, by, you see, normal intercourse, this will not spread. It can still spread from that as well, because there the body fluids, they will be mixing with each other, right? The semen is the male body fluid, which may contain what? The pathogen in it, right? And the lubrication produced by the, you see, vaginal lining, that is also the body fluid, right? So by the mixing of this, it can spread. Now, another thing that you see, spread can also happen because of the sharing of the needles as well. Now, you guys know that, you see, the junkies or the addicts people, they use, you see, this heroin, they inject heroin. So they are already poor, right? And they are buying drug because, you see, they are you see, taking alms, you see, begging for the money, and then they are buying the drugs, right? So they don't have money to buy the separate needles for themselves. So what do? That they're sitting in one place and they're sharing needle with each other. Let's suppose one of them was HIV positive. So what will happen? When the needle will be injected in the blood vessel, from the blood, the pathogen will go on to the, you say, this needle, and when it will be injected into another person, this HIV is, you see, transferred. It can be transferred from the barbers as well. Now, people, they go for shaving to the barbers, right? For having haircut or something. Now, let's suppose that a person goes to the barber who is HIV positive. The barber is shaving him and gives him cut, right? Now, the blood will come onto the blade. Now, if the barber is cheap and he do not throw that blade away and does not sterilize it and uses the same, you see, blade onto the next person who is healthy and gives him the cut as well because it's cheap, right? So what will happen? He will be transferring the pathogen from one person to another person. Another, remember one thing, that the HIV does not spread by talking to someone, right? It only spreads or shaking hand with someone, or hugging someone, the HIV does not spread like this. HIV will only spread when the body fluids, they will come in contact. Understood everyone?